It is for real my honor to introduce Changson. And for those who don't know him, um, the thing is that I think it will take a minute, so just prepare. It's a lot of things to say here. But yeah, Changson is a chair professor of physics and complex systems in the Department of Physics, Hong Kong Baptist University, but also director of Center for Nonlinear Studies, also director of the University Central Research Facility Life Science Imaging Center, also director of Institute of Computational and Theoretical Studies there, and an RGC Senior Research Fellow. Um, but then also, you see, it doesn't end. Also, he received the university's President's Award for Outstanding Young Researcher in 2011 and President's Award for Outstanding Performance in Scholarly Work 2021. Okay, with all these, how did it even happen? Because Changsung is interested in analysis and modeling of complex connectivity and activity in neural systems using physics, very interdisciplinary work. And as you can see from the title here, it's going to be a wild ride, I think, and I love it. So with that, I'm very excited to hear your presentation, and the floor is yours. I think we can just officially start and cut everything before this point out.
Okay, thank you. I would like to thank everyone. Should be good afternoon, right? Here, evening. Good afternoon, everyone. I would like to thank Kaison and Petra for the kind of invitation to join this Making Connections seminar series and to introduce our work on hierarchical modular organization in the brain, which enables the brain to organize segregation, integration, and balance to support diverse cognitive abilities. So here is an outline of the presentation. I will start with an introduction, basically the concept of complex system to treat the brain as a complex system and also its general direction of our research. And then we'll come to this topic of hierarchical modular organization in the segregation integration balance. And finally, will be a very brief application of this framework or method of detecting hierarchical modular architecture to like stress and patient or disorder situation and finishing with a brief summary. So here is the introduction. So generally, I'm interested in understanding the complexity of the brain because, like AI, our brain is doing this response to the stimuli and generate behavior outcome, but they're very different from a typical like AI system. We have a very complex intrinsic states, and that is already a lot of activity even without doing task at resting state. And importantly, this kind of resting state will consume about 95% of our very valuable brain energy. So this is a very big puzzle or interesting question that how this kind of complex dynamic is generated and is supporting the function. And in typical like a neuron cognitive study, actually, this kind of activity is usually treated as a stochastic background and noise. But people like us from physics background, from a complex system, we believe that this is a largely like self-organized dynamical activity and has important functional meaning. So our research is basically very much interested in understanding the organization principle, dynamical mechanism, the functional relevance of such complex brain activity. And this is kind of motivated from this framework of the complex system. The complex system saying that a system like a neuron system with many nonlinear elements like the neuron, and when many of them are coupled together, then we have the expectation of the emergence of a complex activity like a neuron system. The individual neuron spiking, I will show you later, is a very irregular, but we can have a collective emergent activity like oscillation and criticality I will emphasize. And over the last 2 or 3 decades, and we have a lot of emphasis on the connectivity, the network neuron science, emphasizing on the complex interacting pattern among these elements. And also, as a complex system, there's another kind of dimension is about the diversity or heterogeneity, means like these units we are talking about is not uniform. Actually, it's diverse. It's like we now can map out the different types of the neuron in the animal brain. And when you come to like human brain, come to the level of the brain network of different brain areas, and we know that these brain areas is not uniform, has this hierarchy of anatomy, physiology, and also gene genetic expression profiles. And when we talk about the connectivity, like this structure, and one very important feature today I'm going to emphasize is this kind of a modular architecture, or you can say the brain network is a network of a network. Apart from those complex network features like a smaller and scale-free module, and here I emphasize the module and later the hierarchy, because you can see like when we go to look at the brain and it is divided into a few like functional subsystem, but within this functional system, the connection, this long range projection connection among this brain area is denser, and then between the functional system or between the modules, it is sparser. And here, I think when you look at this kind of picture, you have first question maybe is whether there's a kind of balanced distribution of this connection within the module or between the module. And that is the key question I'm going to discuss today, the segregation and integration. And when we talk about the activity, I mentioned the self-organized activity. And a very important feature of this brain activity, from especially from a physical science perspective, is the avalanche or self-organized criticality. Uh, so when we put electrode into our brain to measure the spiking of the neuron, we found that is actually very irregular and similar to a Poisson process. But many neurons can spike together, and then this kind of a clustered spiking is called avalanche. And if we look at the distribution of the size of the avalanche, it has this kind of power law-like distribution in the size and in the duration, and then between the duration and the size, there's also a scaling relationship. And importantly, this exponent of this distribution, power law distribution, also follow a relationship. It's called crack noise relationship. And all this evidence suggests that the system is closer to a critical point. And this kind of hypothesis that brain is working at the critical state seems very general. You can see in different species and in different states and in different scale. For example, if you look, you can look at the avalanche at the electro level, this local field potential level. And when we come to the whole brain, and our human brain is a very big one. And nowadays we have a lot of study network neuroscience look at this functional connectivity from the perspective of functional connectivity. But if you look at this kind of activation cluster or avalanche, and you see that it has a very clear evidence of this criticality or power law, and with a very strong variability of this kind of a cluster size. And this evidence shows that our brain seems to work at this kind of critical point, at the transition point between too little or too much activity. And so this kind of activity, ongoing activity, use up a lot of energy, and later we will understand better, I think. This is because this kind of state provide a very sensitive standby state for efficient processing and and the flexibility of our system. Because from statistical physics, at a critical state, the system is very sensitive to perturbation. And at the end of the talk, I will show you that actually this idea like the brain is working at one critical point is not completely correct. Actually, there's a broad range of critical states called graph phase. So I just mentioned this flexibility or sensitivity. Actually, you have a lot of evidence. Like when a neuron circuit is working at the critical state, you can have the maximal dynamic range, means the system can have a response to a very small perturbation or very large perturbation. And these different avalanche combinations, if you think of possible representation of the information, then it has a maximal information capacity. And in the neuron system, people often talk about synchrony, but synchrony is not static in neuron system. Actually, you have a very flexible or maximal variability of switching this synchrony or coupling or decoupling. So all these provide the evidence like the brain is a very flexible, very sensitive. Actually, we have a recent work to show that at the critical state, actually the system is also most reliable. Means you can have a repeated response to the same stimuli if you repeat input. And in our brain is a functional system, has the ability to perform, to conduct many tasks. And these different tasks, performance could be correlated to reflect the latent ability, like a fluid ability or crystallized ability, like reaction speed, processing speed, and all these different ability actually has a correlation which is explained by a common manifold. It's called the general ability, the G factor. So this is a kind of hierarchical architecture in our intelligence. So basically, as Kaison emphasized that we are interested in this very complex brain dynamic and cognition and trying to build this kind of multidisciplinary uh, uh, research template, trying to integrate this nonlinear complex network approach for analysis and modeling with this cognitive neuroscience approaches for experiment, especially emphasizing on this complex brain dynamic or variability, which has been largely treated as a variability or background fluctuation. Because this dynamic is a bridge like linking the underlying network architecture of physiology, which can change across individual or can be changed by disease, to the function, which also have a variability across individual, aging, or disease. So by studying this complex dynamic, we hope that we can obtain better understanding of the structure, function, dynamic function relationship, and based on that, we may develop some new algorithm or software that could be uh, useful. For example, for detecting dynamic based like a sensitive biomarker for early diagnosis or prevention of brain disorders. So following this line, and we have been working on like different scales of the brain system, like a local circuit model to understand these different dynamical process. For example, model can unify this irregular spiking, oscillation, avalanche, and in a relatively large scale, you can observe this propagation of the brainwave and how you can have a local or global critical state. And when we come to this whole brain level, then we can integrate this imaging data, functional imaging data, and here we can discover this kind of organization principle of hierarchical modular structure in organization in the structure of functional connectivity, supporting this different cognitive ability with individual difference. And this is the topic I'm going to talk about today. And overall, what we are doing is we treat this complex activity as a network-generated dynamic and to characterize the brain states or to understand the mechanism of information processing. So this is a kind of overall introduction of our computational work. And as Kaison introduced, we also uh, promote in our university to establish this life science imaging center with this system level brain imaging like MRI, EEG, TMS, FNIR to support the transdisciplinary uh, brain science study across different topics, yeah, disciplines. Now, after this general introduction, let me come to this topic of hierarchical modular organization, the key topic of today. So when we talk about segregation and integration, I think the neuroscience, the traditional view is like the brain can be divided into specialized functional areas, and then these areas are supposed to do different processing, and finally we have the at the higher order of the hierarchy, you have this integration. And this is still very important to underline, like a lot of imaging study trying to localize these different areas underlying a cognitive process or task. But nowadays, we also realize that the one brain area is actually involved in many different functions. And this is already like suggesting that it is working as a network. So the network neuroscience actually in the last decades went to, I would say, the other extreme by somewhat ignoring this region specialization, but emphasize about this interaction among these regions. Then when we have this network, what how can we understand this so-called functional segregation or integration? I think a very natural picture is this kind of a modular architecture, because when you have a module, we have a stronger connection or denser connection within the module and a sparse connection between the module. This means we can have relatively specialized processing within a module without being too much interfered by the other module. This is specialization. But then still we need to have an interaction among the module in order to realize this higher order interaction. So again, from this kind of illustration, you will have a question like how we somewhat distribute this connection between the module. But then this is the a picture like seems you just have one level of module, and I'm going to emphasize that there are many levels of module architecture in the brain. So this kind of question is actually starting from the first day, like I came to this field exactly 20 years ago, more than 20 years ago. And when I come to work on this cortical network, and we ask the question, what are the special organization features? What is the relation between the structure of functional connectivity and how the system can realize balance of segregation integration, like together with Gorka. I'm not sure whether Gorka is only on the talk. So at that time, we even don't have a direct comparison of a structural or functional connectivity from the same species. So we already built this so-called nowadays large-scale network, like each area with hundreds of neurons and coupled through this catechortical network. And our focus was to understand how this structural module is going to constrain this functional module, and we found that the yes structure can have some constraint on the function, but the function can go beyond the structural modules, and that is the thing I'm going to emphasize further. And and when we talk about the module, actually, there was at that time we already have this kind of picture, like this kind of hierarchy, because we found that in the cortical network, when you look at a few functional modules, and we found that some of the area from each of the module, they form a kind of what we call hyper community. Later, people borrow this term directly from this social network rich club, and this area has a kind of very highly dense connection and overlap on these functional modules. And we found in another species, this macaco monkey, and this hyper community also absorb most of the long range connection to link this spatially segregated functional subsystem. And that is a kind of very cost effective organization, because those cost a long range connection is concentrated, and then you leave most of connection is just in the neighborhood and allow this functional segregation. If you have a random long range connection, then you will destroy this kind of functional segregation. And we talked already a few times this module architecture in functional connectivity, and in functional connectivity, people already observe that this module architecture is not static actually, depending on the condition like processing. For example, in working memory, in-back would need to recruit a larger system interaction, and then the modularity is reduced. So when we look at this very important pronounced feature of the architecture, like modular architecture connectivity in those structural connectivity and the dynamic in the critical state, I think the first question we found interesting to answer is in the real brain, when these two are combined, what could be the functional implication. And I'm going to show you that the combination of this hierarchical modular architecture and the critical state, they can maximize this functional diversity and to support this nested segregation and integration across different levels. And these we obtain by combining a kind of very simple model of the brain and with the MRI data, and use this physical concept of eigenmode, eigenvalue of the human connectome Laplacian to understand the structure and function relationship. So when we talk about the structure function relationship, we can map the network architecture or the structural connectivity, which here is from the diffusion tensor imaging of the human brain, is the adjacent matrix. And then you couple these different brain areas through this simple linear, Gaussian linear model. Actually, you can have a much more sophisticated model, but this linear model can already give us a very important understanding. And then the dynamic is just activated by this Gaussian noise, and through the coupling with this coupling strengthening, you can have the correlation emerging from this interaction. And this is called the functional connectivity from the model. And then that is depending on the coupling strength. And then we can compare this model functional connectivity with the real brain connectivity. And through this kind of modeling, we understand uh, why the brain would like to organize at the critical state. I'm going to show you next. So first, in a data set we use a public data set, we reconfirm that the brain is working at the critical state. We look at this avalanche distribution from different resolution, like a voxel level, the thousands of regions or hundreds of regions, and you see this very beautiful power law distribution. Actually, if you look at the avalanche size, duration, and this scaling of size and duration, you realize that this critical state is obtained through this scaling relationship across different scale. Now we come to compare our model with the data. In the model, when we increase the coupling, and initially when there's a zero coupling, then there's no correlation between the areas. And then when you increase coupling, the average correlation between the area increase with coupling. And when the coupling comes to this special value, the dashed line, then the average correlation come to coincide with the average correlation from the real data. This green line is from MRI. And at this coupling point, our model functional connectivity become very similar to the functional connectivity. The difference from the real data, the difference is minimized. And also, our model at these special places also shows this kind of avalanche if you measure their synchrony clusters. And then this power law distribution of the real data and the model also become minimal. So you can see that the brain is compared to the model, the brain is working at this kind of transitional state, if you can imagine a dynamical process. So in the evolution tried to put our brain parameter at self-organized at this kind of critical states, right? But why we want the system want to be here? So here we want to go further to understand from this physical concept of eigenmode. The eigenmode is the picture like or kind of very natural concept to link the interaction with the dynamic. So here is an illustration that if you have two spheres coupled with a spring, and we have oscillation. The coupling is the adjacent matrix, and then the dynamic or the interaction is reflected by the Laplacian matrix. And then when you do the eigenvalue decomposition of this Laplacian, you find that we have two fundamental eigenmode, and this is the motion eigenmode. This is the motion like the two block can the two sphere can work together, move together, or in the opposite way. Then the typical dynamical process or the motion can be decomposed as the combination of these two. So this concept is a fundamental in brain in physical science like statistical solid state physics. When we know the interaction crystal architecture, we already can use the mode of oscillation to understand a lot of property of the solid states. And then this concept has been extended to the connectome of the brain. And then this mode means the activation pattern in the system. So this is closely related to like cognitive science, like how brain areas are activated together. And then these true brain states can be expressed as a combination of this eigen pattern. And eigenvalue means the reciprocal uh, uh, of the lifetime of the mode. So now I'm coming to show you that basing on this mode, we can have this so-called nested spectral partition method, which can apply to the Laplacian matrix of the structural connectivity matrix or the functional connectivity matrix directly. So here is the illustration using the functional connectivity matrix directly and from the average human connectome project. And so here you have a 300 brain regions with certain labeling um, method. And then you see some kind of module architecture, but it looks quite complicated. Now we do this eigenvalue, eigenmode decomposition. And you see we look at the ordered according to the eigenvalue and descending order. The first eigenvalue is very large, and then the eigenmode looks like this. And it's almost flat, means the whole brain is activated together, up and down. And then the showing that it is whole brain is synchronized. But if you look at the second, third mode, you have some positive and negative, and it looks complicated. Positive means activate, and negative you can think of like deactivation, and some area are activated together or not. So you don't see too much, but since the sign of the component of the eigenmode is indicating of activation, then we came to the idea that we can look at those areas with the same sign, with the same direction of activation, and then we group them together. Means we shuffle the label. And then when we put them together, shuffle the label and go back to the original functional connectivity matrix, and you see this very interesting emergence of a module. And then the coupling between the module is much sparser. And now we found a larger module and a second mode. And then based on that, we go to the third mode. And this means within this module, we again look at those areas with the positive or negative eigenmode activation sign of this component, and then group them together or reshuffle them. And then you can see that in the original matrix, we see this emergence of a smaller but a denser module. So this is a method like you can apply further and further. That's why we call nested. And then up to the level that you divide the brain into a single area. And then you can imagine that going back, and two areas are formed a smaller cluster and four areas, and sometimes like more than two areas. And then you can see that there's a hierarchical module architecture, and this is called nest spectral partition. Now let me come to the structural property of the network. And so we apply this to the adjacency matrix from the DTI and translate it into Laplacian matrix, do this kind of hierarchical decomposition. And the first mode is the flat, and I don't show the second mode. And when we organize according to this positive and negative sign, and we found it's very naturally corresponding to the hemisphere, the left and the right hemisphere. And then we go to the next level, and then you can see it is dividing the brain into the parietal and the frontal largely. And then if you go further and further, you can have a finer division of the system into smaller modules. And this kind of architecture showing that the connectivity already allow certain mode of activation. For example, this segregation of left and right, and actually you can also realize somewhat in a suitable condition. For example, when we travel, we have the so-called first night effect. Half of the brain can be more activated than the other. And now, after we reorganize the label according to this functional hierarchical module, and we go back to look at the connectivity, the actual, structural connectivity. And when we relabel this area, you can see the underlying structural connectivity now reorganized into this very beautiful hierarchical module architecture. And this kind of hierarchical module in the Laplacian, because underlying structural connectivity has this hierarchical module architecture. And this kind of a module architecture is a very robust and with respect to the parcellation resolution. If you go to like a final parcellation of thousands of area, you can see largely the same organization, but certainly you can get smaller and smaller more levels of the hierarchy. And if you go to more coarse scale, and you still see this kind of hierarchical module architecture, right? And I'm going to show you from this kind of a module mode decomposition to understand why the brain would like to stay at the critical state. And here the concept is we measure the diversity. I will show you what is diversity or complexity in the functional connectivity. And we found when we come to this special coupling, when the model is at the special coupling, that we can recover the functional connectivity from the real data, and we found that like this diversity is maximized. And from the model, this diversity we measure, it is very close to the real system from directly measured from functional connectivity in fMRI data. And at this critical state, it is special because when we increase the coupling, those small mode, you can see those very dense local area become synchronized, and then you see the when you increase the coupling, larger and larger mode start to emerge, and this special critical state is the place like a few leading mode become maximally activate, and then this means inside the brain, when you have this hierarchical architecture, will give you this mode, but these modes are best activated or used at the critical state and to give you the very diverse combination and give you the dynamic. So what do we mean by diversity? So here is an illustration when you couple this system, when the coupling is very small, and you can see the interaction between the area is very weak, and the correlation between two area, and if you make the distribution of this correlation value across the whole network, is very much concentrated to like a near zero distribution, so very narrow. So this system is overly like segregated because it's still like working as a separated area. But if you go to the other extreme, when the coupling is very strong, then the correlation value is distributed close to 1. This means the whole brain is kind of synchronized as a unit. And you cannot allow these different units to work separately. So if you at the critical state, in between what you see is some of the connection is weaker, like between the module and those connection within the module is stronger. And then we have a very broad distribution from 0 to 1. This is from a model, and the real brain is looks like this. You have a very broad distribution of this uh, correlation. So this distribution, the broadness of this distribution, we call diversity, and which is maximized, and as I showed you. And this kind of diversity is very importantly, depending on the hierarchy. Actually, we did this kind of control. If we go to a module and then we can destroy this module architecture within this module, just randomize the connectivity, and then we just get like this two level of hierarchy. But we could go to some fine scale and try to randomize the connection within. And when you do that, you find that when you, for example, randomize the whole network, you lose this kind of diversity. And in the real brain with this hierarchical module architecture, we have the highest diversity and also the broadest parameter that can give you the high diversity, means the system is very, very robust. And um, so this is a kind of evidence to show you that why the brain want to organize into in this hierarchical architecture. Now, if we use this method, and you can apply, for example, to default mode network, you can further divide the system into different levels. For example, for default mode network, with this division, we can divide into this so-called posterior and anterior default mode network. And in different studies, people already found that these different subsystems can be recruited in different cognitive process or being targeted by disorder separately. So here is a short summary. It seems like I am a bit slow. So the brain is overall self-organized at the critical state, which have come from maximal diversity, and the hierarchical module organization give a very high capacity to support this diversity. So this shows that the brain has the evidence of evolutionary optimization jointly in structure and function. And you can imagine that when we go to final partition, and this hierarchical module architecture can go down up to like a single neuron level, and even go to this level of single neuron. You have different types, and you have many levels. Now let me come to the second part. Let's talk about the segregation integration in a functional network, and how we can more to quantify this kind of segregation integration. And is there a balance? Like this is the question I asked from the first day, and how they could be like associated to the cognitive ability. So again, we use this method of modeling MRI data. Now we also go to the cognitive data, and also go to this different ability using the structural equation modeling. So this concept of segregation integration has been discussed in a kind of quantitative way, like from psychology, and then we can see that some states is easy to realize and some integrative states is difficult to reach. And I just showed you like from physical science perspective, and we have a transition, maybe close this different hierarchy of the module architecture. And then now I'm going to show you that with this kind of hierarchical module architecture, we can have a way to characterize this balance. So here is the application of this nested spectral partition to the functional connectivity. And we go to the first eigenmode, which is uniform. The second mode, you divide into negative and positive. Now, very interesting question, interesting that we still don't understand well, is earlier in the structural mode is the hemisphere. Now it is not. The second mode is more like a frontal parietal system. Actually, is the default mode and then the other part of the system. And then you can go down to separate into hierarchy mode, hierarchical functional module. So the first mode is actually indicating the whole brain is synchronized, has the largest eigenvalue. So its square of the eigenvalue is much larger than the others. So we can use the square of the eigenvalue as the energy or contribution of this integration, the global integration. So we call it integration component. And then the other component, you divide the brain into different levels of the segregation. And you can see segregation integration is a kind of a relative concept because you can talk about integration of a smaller system into larger system, and or you can see segregation into smaller and smaller. But we try to look at these different components of segregation and add those eigenvalue square of eigenvalue we call segregation, and the difference we call a balance. Then we found very interesting is again in this model, we can have a coupling value that you can recover the functional connectivity as most closely to the real brain functional connectivity. And what is special we found is at that special coupling, when you increase the coupling, the integration component is getting higher and higher, and the segregation component becomes smaller and smaller. And at that special coupling, when the model match with real brain, we have this crossover and we have the balance. Then the difference is zero. When the model can calculate the infinitely a very long time series. And from the data, the fMRI data HCP, we also can put the data together, very long time series. And we found that this measure of segregation integration is almost zero. And in the real data, it is biased to segregation because you have a lot of noise, and we have this noise component will give you some eigenvalue, and so it is biased. And so overall, you see this balanced in the healthy young adult. But if you go back to the graph series, people use like a modularity of this participation coefficient, and you don't see any special at this like a critical state of the brain of balance. So here is a measure like you can directly talk about the balance. So when the brain is at the balance, actually, if you go to measure this for a different individual, and we found most of them are actually this each dot is the individual is at this critical state or balanced state. And when the average brain here is a real one of the real brain is on average is balanced, and you can see over time our brain has a very flexible transition across different segregation and integration. If someone's brain is overall segregated, and you see it is very hard for the brain to have transition to synchronized state or integrated state, or the other way, if the overall brain state is integrated, it's very hard to go to the segregated state. So at the balanced state, you have a maximal flexibility to have this dynamical transition. It's like the brain is monitoring the state and have a standby, and when external signal come to require you have segregated processing, then our brain can switch to or if you have integration, and then can switch to this state very quickly. So therefore this kind of balanced individual difference in the balance can be treated as a trait of our brain, and then individual trait, and then we expect that that can predict our cognitive ability. So when we talk about cognitive ability, these different tasks can reflect different latent ability, and we use this structure equation modeling to associate resting state brain measure to the behavior, and we found that we can explain them. But we don't find in this linear model, we don't find the relationship for the memory. And actually, we found that because there's a nonlinear relationship, so the memory has the best performance at the resting state. But for like the general ability, if you divide into group, and because general ability means you can do many tasks, and you need many of them need the interaction or coordination of different brain area. So you require a higher integration. But for crystallized ability, then you need a more segregated brain. So overall, when we have a complex, diverse pattern, you have kind of a trade-off of the segregation and integration. And now a few last slides, I want to emphasize that the segregation integration is indeed associated to the critical state that I showed in the first part. But we want to enhance this concept that the brain critical state is not one point because the brain area is heterogeneous and the network is modular and heterogeneous. And we found that the combination of regional heterogeneity and the network heterogeneity can generate a broad range of critical state, and this is called the graph. And this different brain subsystem has different uh, uh, transitional properties. Some is biased to subcritical and some is biased to supercritical. And but overall, the whole brain is in a critical state. And very importantly, we have a model to calibrate this brain data to show this power law distribution of the avalanche happens not in a homogeneous model, just at a one almost like a very narrow range of 1 point, but in a heterogeneous model like the real brain, is a very broad range of critical state. And this is in the model and also in the real functional MR data. And so the real brain has a broad range of critical state. And so what we found is a typical individual. Everyone has this kind of critical state, but only a few small percentage of individual is going to some extreme of a subcritical or supercritical states. And then these critical states is exactly the states that we found that we have balance of segregation integration and we have the maximal diversity in the functional connectivity or maximal flexibility of transition cross segregation integration. And again, and you can predict this different ability profile. So now I used to 1 or 2 minutes to summarize or to show you that this kind of hierarchical module measure of segregation integration. Actually, we can go to each area to see how this area is more segregated or integrated from the rest of the network. And we apply to a state reconfiguration in the acute stress. Under stress, for example, today I'm going to talk and I feel a bit stressed, then my brain become overall more integrated. And you can identify those like a stress sensitive area, which we found that is more robust than this network based method. And then you can also see the brain become more stabilized, or the variability is reduced and with the stress. And we can apply this method, for example, to a disorder like this lifespan of ADHD. And we found that this integration is overall increasing in the normal population linearly, but in the disorder population, there's a kind of non-linear change of the segregation integration. And this flexibility can also reflect the different system. In different system, we can identify those different systems can predict these symptom subtypes like hyperactivity or inattention. And then that's the basically the evidence to show like this method by looking at the co-activation of the brain area, which is naturally linking to the cognitive process of the brain, can apply to cognitive function or disorder. So now I come to a summary. I showed you that this kind of evidence like a brain structure or functional network display very pronounced hierarchical modular architecture organization, and there's evidence of optimization in both structure and dynamic. And this mode-based method seems very naturally linking structure to function and is interpretable and is very powerful, I think. And then many of these like age-based like graph serial method and because we also do not need to do thresholding. And we found that resting state of young adults are on average at the critical state and at this segregation integration balance, and they can predict the individual cognitive ability because this is a individual trait. And this modular architecture is a kind of crucial for normal and diverse function, but actually it is also a vulnerability for neuropsychiatric disorder, and I don't have too much time to show that. I didn't have time to show you, but we have work to show. Actually, a lot of weak connection in the network neuroscience are ignored and threw away, but they are very important for this segregation integration, especially to keep the balance of segregation integration. And overall, what we can see that the seems hierarchical modular activity, connectivity, and this kind of critical dynamic is underlying our human nature, natural intelligence. And that I think is also important to inspire like human like AI. So with this, I would like to thank my major collaborators. Dr. Wang Long is now from Xi'an, and my former student now is a PI at the Shanghai AI Lab, and our collaborator is an expert on psychometrics from Germany. And I also would like to thank you for your attention. Anyone interested in visiting Hong Kong or even studying with us, there's a very prestigious like Hong Kong PhD fellowship. And thank you very much. I kind of overused some time. Yeah, sorry.